Steve, you have a comment question?
Yeah, I had a question, Aditi. From your readings, have you seen a good explanation for once you get the spike protein expressed on the surface of your own cells through delivery, either from the lipid nanoparticles with RNA or the adenovirus with DNA that's translated into RNA and then made into the protein? Have you been able to read where there's been any real explanations for the steps that are involved after that to actually getting, T helper cells activated or B cells ultimately in the lymph nodes? They're injecting in the muscle in the arm. What have you seen as been the explanation for how the vaccine actually works?
I'm not sure anybody really has done a systematic study to see how or where the mRNA is going, because in the biodistribution studies done by Pfizer and Moderna, they never actually looked at the mRNA distribution. What they looked at was a surrogate, which was basically a vector, which is
I got that. No, I'm assuming that it works, that the spike protein, as a trimer, is on the surface of your own body cells now. So how does it get from there? What's the steps that you're going to now be able to get an immune response of antibody production?
Well, I mean, that's one of the biggest questions. That's why I went through those whole steps of showing you how your thymic education works.
Right.
And it's not just for spike protein. For no other vaccine in the past, it's been shown that those can elicit that thymic education. And that's why.
Right. I got that. But in those cases, you've got a vaccine that is small. The either it's a attenuated virus or even as tiny as a fragment of a protein, a peptide, and your neutrophils or macrophages or dendritic cells, they engulf, digest, and present with major histocompatibility antigens to then educate, the T cells. But how do you have it when your cell is much, much bigger or the same size as an immune cell that's presenting this foreign protein on your own body cells? How can that be achieved?
Well, so there are two things. One is that I'm not sure if anybody's actually shown that people who are vaccinated have this, it's an artificial system where they look at their spike antibody to the receptor binding domain in a very artificial assay. So but I think one explanation is that the spike protein actually gets shed, just like if the virus had the spike protein, and it gets into these exosomes, which are more like also lipid particles, which your body, so they will sort of pinch off. And those exosomes are the ones that make it to the lymph nodes and then are presented to those immune cells to then make antibodies.
Right. I guess the point, sorry.
And the other is that, of course, if the RNA is not able to, if it's degraded, it's not going to make a protein. And then the RNA itself is a non-self, right? So then it can be eaten by those macrophages or neutrophils, just like if you had a viral infection, because again, whether it's the mRNA vaccine or the adenovirus, they're all non-self. They're not your DNA, they're not your RNA. So your body will recognize that, and then they can be engulfed, and they may then be able to elicit some of the innate immune responses that you would see with a regular viral infection.
I suspect you're right that it's the generation of exosomes that where you have fragments of the cell membranes that have the spike protein on them. They're small enough that they can be engulfed by antigen presenting cells. But in order to generate those exosomes, I suspect that there's actually an inflammatory attack where your immune system's coming in and causing that damage and harming the host cell that's expressing the spike protein. And that may be because you have some sort of an immune reaction from cross-reactive proteins against other coronaviruses or whatnot to start it. But eventually you get all this infiltration of these immune cells that are attacking your own tissue, essentially, generating these exosomes that then get taken up by antigen presenting cells. But the bottom line is you're also going to be presenting a lot of parts of your own body proteins. And although you may have tolerance, I would suspect that at high levels, you'll start to make antibodies where you may in fact get cross reactivities that then come back and start inflammatory responses later, even in the absence of the spike protein, which is really the beginnings of autoimmune disease. So I think it's really important to understand, how does these vaccines actually work? And to my knowledge, it's very fuzzy. And this is what I would suspect. And I don't know how you feel about that.
Yes, I agree with you that these vaccines the RNA and DNA vaccines may lead to more of autoimmunity, also because, like you said, unlike the viral vaccines, which, the dead virus doesn't replicate, right? So once you give a bolus, whatever you've given is what your body will see. Your immune cells will engulf them and then either evoke an immune response to give some kind of antibodies or may not, depending on, and basically then you're done. But with these RNA and DNA viruses, especially, the RNA, the thought was that this mRNA would get cleared away and your body will not be constantly making spike protein. But there have been reports where I've seen that 60 days after the first injection, people have been able to isolate mRNA against the vaccine, so the vaccine mRNA. So the question is, for someone who works with RNA and to be able to make so much RNA that you can sequence from your blood draw, makes me think that if you gave them only 30 micrograms 60 days ago, why wasn't that eliminated? And the 30 micrograms is now getting diluted in over 1 point whatever liters of blood that you have. So it's not like you have 30, if I draw 1 ml of blood, there's not 30 micrograms of mRNA in there. It's diluted several thousand-fold. And now it's 60 days later. So how can I get so much RNA without it being amplified? So is some amplification of that going on? Why is it not degraded, not getting cleared away? So that's a concern. And if that mRNA is hanging around, then obviously it's being targeted to different cells and different people. And depending on the cell type, unlike immune cells that are geared to make only one kind of or only a very select kind of proteins, because you're going to make antibodies to, your other cells don't. They have to work with making thousands of proteins. And so now if they get overwhelmed with having to make one kind of protein, which is not really in their normal repertoire, then you will have a price to pay. So I think, and the DNA adenovirus adeno-associated viral vector, which is used for Johnson & Johnson and AstraZeneca or Oxford vaccine, that's equally scary, because even though they take these adenoviruses that are supposed to be mutated so that they don't integrate in our genome, but we, there are populations that have natural adenovirus infection, and they can perform a rescue experiment, meaning they can provide the missing part. So it's like saying that if you have a car that lost its tire, and now you can just take a spare tire from another car that's also there. So can that happen? Of course that can happen. And that's why FDA had this requirement that any treatment which was normally adeno-associated viruses were used for gene therapy. And we've seen most of the gene therapy trials have been a disaster, and they've stopped after 4 deaths. And of course, here you can have 4,000 deaths from the vaccine, but somehow this vaccine is untouchable, like I said. But there's also no requirement of follow-up for 5 years, which is just amazing. So there's a lot of shortcuts, sloppiness, and if the scientific community doesn't speak up, I just don't know.
Yeah, Didi, actually, there's been a number of studies with cell culture that shows that when you have transfection, basically the introduction of the RNA, that with these vaccines, that with the lipid nanoparticles, it actually induces the production of an enzyme called a reverse transcriptase, where the RNA is actually converted back to DNA, which is much more stable. And then you get continued production. This is in liver cells, for example, which is one of the main sites that the vaccine travels to after the site of induction in the muscle. And there you can start to continually produce more and more RNA because the DNA is much more stable than the RNA is. So that's probably the mechanism by which, even a month later, you're still measuring, very high levels of RNA enough that you can amplify it and detect it.
So I've seen that paper
called line 1.
Right. So line 1 doesn't, in that paper, they actually don't show line 1 transcription happening.
There's another paper that reproduced that data that was published.
But there are other enzymes that we actually have, which are called DNA theta polymerases. And they also have a reverse transcriptase function. And for some reason, they seem to gravitate more towards the, instead of doing the DNA transcription, they actually seem to gravitate towards viral or pathogenic mRNA and take them and do RT or reverse transcription. So it may be that there are mechanisms. Whether or not line 1 is involved, I'm not so convinced, but there are other definitely mechanisms that can show that, can do that and have stable RNA converted into DNA. But the fact is that they are still detecting this viral mRNA vaccine in blood, and that's not what's supposed to happen. So saying that whatever it is, we need to pause and take a look or to see whether or not these mRNA vaccines are actually safe or not. So I'm just looking at, sorry, some of the comments, and I see that you said that, yes, there was the prevailing wisdom at that time to take out tonsils and things like that. And there's been a study that actually looked at going back 60 or 80 years and following up, and what they found is that for whatever indications they told them to take out, whether it was snoring or difficulty breathing or whatever it is, those things don't really change between people who have their tonsils taken out versus don't. So whatever it was meant to cure, it doesn't really cure that, but it ended up giving or increasing the risk of more respiratory illnesses for people who don't have tonsils and adenoids or appendix and things like that.
Actually, the funny thing was that your explanation given at the time was that when you're sick, you feel pain in your neck area, and this was to reduce the pain, much like we try to reduce temperature when someone's sick, when in fact we're reducing our immune function because it works better at higher temperature. these are natural responses of the body. The idea was simply that the doctor said, well, cause pain, let's just take it out. And that's
Right,
what,
and that's another point to say that, obviously, I think maybe Barry said that he tried to convince his patients or not to get them out. This whole thing of that, if you don't understand what this is doing now, it's either vestigial or you don't need it or whatever. So if we don't understand in that time and in that context, that's it. There is no other function, right? And that's the, I don't know, snobbery of science, or we think that we know everything. I don't know. And then once you've done that, it's very hard for you to admit that you were wrong. So now if everybody else has adopted it, then for the whole community to come out and say, I mean, look, we have the same thing with antibiotics, right? Before antibiotics were these they're still wonder drugs for what you can do, but that doesn't mean that you just keep popping them, right? So, I mean, there is also this thought that your appendix actually, and in animals, it's cecum, right? That is the reservoir for your backup bacteria. So if you have an infection, then your gut loses that bacteria, but it's very hard to get into the cecum or the appendix. So then it draws, once after the infection, it needs to repopulate, then it draws on the bacteria that it's keeping in the reservoir in your appendix in humans and in cecum in animals. And if you remove it, obviously there's no function if you're healthy because you don't need to repopulate it. But when you keep, from time to time, when you get these GI diseases where your bacterial population is compromised, then that's when you need your appendix. So it's unfortunate that we don't, once we've made a mistake, it's very hard to go back and say, we were wrong, let's correct it. And it takes time. I mean, people are realizing that in the medical community, there's no more that push to take out tonsils or take out adenoids, but I don't know about appendix, whether there's push or not, but hopefully, we'll just have to continue this fight and try and get some sanity back into this discussion.
I do want to bring it back to the mask. This was a great discussion. I didn't want to stop that. Does anyone have a question for Aditi regarding the mask narratives, or if they still believe that the mask is working? We would love to hear, because maybe we're missing out on some aspect of masking. And I would love to be enlightened with why you think you need to wear a mask, or why everyone else needs to wear a mask. So if there's any questions, I mean, this call is not a judgment call at all. So please ask.